Here's the DNA sequence. The bottom strand is the template strand and the top strand is the coding strand that goes from 5' to 3'. Here's the mRNA sequence going from 5' to 3'. The amino acid sequence starts with methionine and then tryptophan, isoleucine, threonine, cysteine, and then finally ends with the UGA stop codon. The first nucleic acid point mutation is a missense mutation. We are going to insert in an adenine here. This will make the amino acid aspirinine. So a missense amino acid point mutation leads to a change in the amino acid. There are different types of substitutions. The first one is a transition, which either goes from a purine to a purine or a pyrimidine to a pyrimidine. The other substitution is a transversion, which goes from a purine to a pyrimidine or a pyrimidine to a purine. We went from uracil to an adenosine. The substitution here is a transversion because it is going from a pyrimidine to a purine. The next point mutation is a nonsense mutation. Here we substitute an adenine for a guanine. This will result in a stop codon. Nonsense point mutations go from amino acids to stop codons. This will stop translation from completing the amino acid chain. The substitution from guanine into an adenine is a transition substitution, substituting a purine for a purine. Next is a silent point mutation. We are going to substitute cytosine for a uracil. This will not change the amino acid sequence, since silent point mutations code for the same amino acid. The substitution of cytosine for uracil is a transition substitution, going from a pyrimidine to a pyrimidine. Last is a read-through point mutation. Here we are going to substitute a cytosine for an adenine. This will make the stop codon into a codon for the amino acid cysteine and will result in the continuation of translation. The substitution of a cytosine for an adenine is a transversion substitution, going from a purine to a pyrimidine. Thank you for listening.